Hello everyone, welcome to our channel CV Forensics. I am Rekha Bandyopadhyay, volunteer of CV Forensic. Today we are going to discuss about the crucial topic designer drugs and club drugs. From ecstasy to ketamine, and MDMA to GHB, these substances are often misunderstood and can have serious consequences. Let's dive in and explore what they are, how they work, and what risks they pose. Today, first we will see the introduction about the club drugs and designer drugs. Thereafter, we will see the synonyms and examples of club drugs and designer drugs. After that, we will see the analysis of club drugs and analysis of designer drugs. Thereafter, we will see the advantage and disadvantage of analysis and then conclusion. Now we will see the introduction about club drugs and designer drugs. But before this, we will know what is drug. A drug can be defined as a natural or synthetic substance that is used to produce psychological or physiological effects in humans or animals. Now we will come designer drugs. What are the designer drugs? Designer drugs are produced in laboratories. The majority resembling drugs legally restricted for distribution. These drugs that speed up the messages between the brain and the body. On the other hand, club drugs primarily used for youth and young adults at dance or all-night parties and bars. That's why it is called as club drugs because we use these in club. Now we will see the synonyms of club drugs and designer drugs. The synonyms of club drugs are party drug because we use club drugs in party. After that, rave drug. When we take club drugs, we become very excited. That's why it's called rave drugs. After that, dance drug. When we getting excited, we also dance. That is called dance drug. Now, designer drug. We will call it synthetic drug because we synthesize it and then novel psychoactive substance and then custom drug and then laboratory made drug because we made it in laboratory. It is illegal but we made. That's why it is called laboratory made drug. Now we came into the analysis of club drugs. We used two techniques, chemical and instrumental. Chemical analysis means Markowitz test, Siemens test, and then instrumental analysis means GCMS, which is a combined form of chromatography and spectrometry. GCMS means gas chromatography, mass spectrometry, and then FTIR. Chemical analysis of club drugs. Markowitz test. First, we add sample and then we add Markowitz reagent 2 to 3 drops. What is Markowitz reagent? Markowitz reagent is 95 to 98% of sulfuric acid means H2SO4 and 40% of formaldehyde means HCHO. Then if we will see the color changes in dark blue or dark black, we can tell club drugs may be present. After that, we will come into the Siemens test. We take sample and then add few drops of Siemens reagent. Now what is the Siemens reagent? Siemens reagent is the mixture of sodium nitroprusside means Na2FeCN5 plus acetaldehyde CH2CHO and then sodium carbonate means N2CO3. Then if we will see the color changes, turquoise color from black color, then we will tell club drugs may be present. After that, instrumental analysis of club drugs. First, GCMS. It was used for determination of chemical profile of drugs. How can analysis? First, we will take powdered sample means powdered drug, then dissolved in distilled water and mixed with D5 MDMA solution as an internal standard. After that, we add 10% of N2CO3 and then mixing is followed by addition of pre-distilled water and centrifuge. After centrifuge, ether layer separated. transferred into a glass insert of GC, a microvial for automatic sampling and dried. After that, the output of GCMS can be depicted as a chromatogram. Now, we can see the analysis of designer drug. Again, we will see the two technique, chemical and instrumental. Chemical is Markowitz test, Simon test, and instrumental is also GCMS, HPLC, HPTLC, etc. Now we see the chemical analysis of designer drug, Markowitz test. First, we take few sample and then add 2 to 3 drops of Markowitz reagent, and then we will see the color changes is orange. After that, for mescaline, which is also a designer drug, we take some powdered mescaline. After that, we add Markowitz reagent, and then we will see the color changes orange red color. After the Simon test, for methylamphetamine, we take sample and then add few drops of Siemens reagent. We will see the color changes purple or blue color. Now we will see the instrumental analysis of designer drug, GCMS technique. First, we collect sample and then grinding and then extract the active compound. After that, we ensure that GCMS instrument is properly calibrated and maintained. Thereafter, we inject a small amount of concentrate extract of instrument. Then separate the components of the extract using a suitable GC column. Detect the separated components using a mass spectrometer. Then we can acquire that data and store data for further analysis. The output can be depicted as a chromatogram. Now came into the conclusion. The analysis of clubbed and designer drugs poses significant challenges due to their complex chemical structure, rapid emergence of new substance, and limited preference data. However, advances in analytical techniques such as GCMS have improved in the detection of and the identification of this substance. Thank you.